Well, hello and welcome to Raj Sabha TV, watching India Fights Back with your host Rajat Kane. This is a show that gets you latest of all the development across the world and of course India as to how we are fighting this COVID-19 pandemic. In this edition of the show, we'll take up a very important issue of strain of SARS-CoV-2 virus or the COVID-19 and its impact on the efficacy of the vaccine. The experts so far ruling out any alarming situation have cited that possibility of a new variant forming any resistance to existing vaccine candidates is fairly low. Well, let us get in a right perspective that every virus, no matter how potent they are, they form their mutants. At the same time, experts have been cautious to come to any downright conclusion on the new strain that was discovered in UK, and it has led to introduction of set of new restrictions on social gathering in the midst of festival season. Several countries, including India, have already declared a travel ban to and from UK. So how important it becomes to understand this new strain and how careful we need to be in the days to come ahead, and what all is required for us to know about new vaccine candidates and its efficacy vis-a-vis the new strain. To discuss more on this issue, we are joined with three important guests. Dr. Anil Gurtu, he is Head of Department Medicine from Lady Hardinge Medical College, Delhi. Dr. Ram Vishwakarma, he is Advisor CSIR, and Dr. Balram G. Omar, he is Additional Professor, Department of Microbiology, AIIMS, Rishikesh. Well, many thanks for joining us, sir. Let me start with you, Dr. Balram G. Omar. For the benefit of our viewers, could you explain the characteristics of any virus and what this new mutant means?
Now, there are two types of viruses. It can be a DNA virus or it can be a RNA virus.
Right.
These RNA viruses are highly mutagenic. That means they mutate very fast.
Okay.
Chances of mutating such viruses is around 1,000 million times.
Right.
Right. So all the respiratory viruses are usually mutating continuously, and this coronavirus has got a property to mutate. Right, it has got a property to delete or add a particular type of amino acid, or it is called as in the molecular word, it is called as a deletion or addition of any nucleotide sequence. Right, so it has got a high capacity of erring due to evolving phenomena. So it wants to become stable. Right, in a particular environment, it wants to become stable. In that process of evolution and stabilization, it changes its forms many times. So it may be a destructive type of mutation, or it can be a positive mutation. Right. So that is the phenomena.
What do you mean when you say a positive mutation? I mean, before I open up this debate to other panelists, let me understand this phenomenon.
Yes. For positive, I mean to say that it will become stabilized and it will not mutate further, or it may not proceed to the destructive form.
Right. We get your point.
Or negative, negative means that with the deletion of some nucleotide, it may evolve to a form which is more infectious to the humankind or to the animals, wherever it is effective.
Right. Dr. Gurtu, I need to understand from you one thing, like as the mutation by itself means, as explained by Dr. Omar in quite a detail, that mutation itself would mean change of the characteristic to an extent, and of course like the change of a genetic sequence of the virus. So now, let's for a moment forget about COVID. For any virus to mutate, how does any medication have divergence on that, on the efficacy of medication for treating any virus if it's mutating?
See, first of all, let us set the word mutation in a larger perspective to understand mutation in a virus. Mutation is normal It is happening all the time to all the life since 4 billion years ago when life evolved. What we are, I, you, all of us, the human, Homo sapiens, are a product of a trillion mutations that have accreted.
Absolutely. Yep.
Now, out of the trillions of mutations that have accreted and grown over the past 100,000 years of Homo sapiens existence and 60 million years preceding that, only a couple survive in our genome, just a couple of them. The rest have fallen by the wayside. Why? Because if 100 mutations occur, maybe 1 or 2 maximum is of use. And that mutations out of the several hundreds that are occurring in all the life forms, that 1 or 2% or even less, which are of some use, which confers some survival or reproductive advantage to that species, they survive in the genome and they're conserved. Rest all fall by the wayside or are there in the genome as silenced areas, a large chunk of so-called junk genomic fragments. So now what happens, the question to ask is that when the virus or a bacteria is attacked by a drug or a vaccine, that mutation which arises in response to this threat to the virus's own existence, like any other organism, that mutation is an adaptive mutation in the sense that it confers some survival advantage to the virus. Good for the virus, bad for us, that mutation. And that is conserved and that replicates and spreads. And so most of the mutations are junk. This virus itself, coronavirus, has had over 4,000 plus mutations since its advent. And we are still counting. Of these 4,000, a bare 3 or 4 have conferred some little advantage to that portion of the virus, which can be dangerous to us, though good for the virus, the spike protein mutations. The current one happens to be a cluster of 22 mutations, not one, there are 22, a set of them, and we are still counting them. Of them, just 3 have tweaked that part of the genome which translates, which gives rise to a part of the spike protein, which is the key that locks onto our cells. Right. So that is not the whole story. It's just a part, a small part of the spike protein, S protein, which locks into our receptor and gets the virus into our system. A part of it has been affected, maybe 3 parts, which amounts to nothing as of today. Only that it is a pointer to the direction that should it evolve further and further and further, we might be heading into some problems. But as of today, it means nothing other than a watchful awareness over the trends of this mutation, nothing more.
Right. Many thanks, Dr. Gurtu, for your opening remarks. So clearly, as you mentioned, that there's nothing to worry, but we have to be watchful. Dr. Ram Vishwakarma, advisor CSIR, I would like to bring you in the debate, sir. As the scientists and researchers around the world are studying this new mutant very keenly. Now, I mean, at the very outset, I know that we have banned the flights to and fro. That is, of course, the administrative decision. But in terms of the science, how worrisome it can be for any new mutant to occur when we are facing pandemic of such a gigantic scale, sir?
Yeah, so first thing I will say that, like my colleague said, it is not an alarm, but I would say it is worrisome. And I will give you context why it is worrisome. When this virus came, and till today, we have genome sequences of this virus, close to more than 200,000 sequences have been done. And actually 12,000 mutations have been recorded. From when this virus came, the one mutation, which we call D to G mutation, there are 2 kinds of around in the world, the D and the one mostly in India, this part of the world, G mutation. And that G mutation conferred a lot of selective advantage for virus in terms of infectivity and the virulence. That was the first episode in last almost 11 months when we put one mutation. And now what we have these 9 mutations. 2 of them are actually what we call deletion mutant, where a chunk of DNA has been replaced or removed, and there are 7 mutations which are point mutation. And couple of these mutations are sitting at the glycosylation site of the spike proteins. As you know, spike protein heavily glycosylated, and that is the portion which antibodies recognize, the natural antibody produced by our immune system and the vaccine-derived antibodies. So if these glycosylation pattern are shifting now, as I can see from a mutation which is known as N51Y, I'm really worried about this because this sitting at the RBD domain, where the virus is binding with the ACE2 protein. Some number it may change the glycosylation pattern. So I think it should be worrisome for us. And it is indicating that because of approval of number of vaccines, we were getting complacent. So I think there is no reason for complacency, number one. And particularly for a country like India, where for last 8-9 months, we have been using these two antiviral indiscriminately. I think my clinical colleague will know, even people are using remdesivir in the mild patients or favipiravir, very heavy dose favipiravir. Now, these are the two compounds which basically disturb the genome of the virus. They integrate in the genome of the virus. And these drug-driven resistance development is also a mean. So I think, to me, I would say that we cannot as a normal mutation, because you see the data that is coming out from UK, this mutation, this set of mutation, what we call new phylogenetic cluster, which is emerging, which is nomenclature given 1201, this one is giving 70% more advantage in transmissibility of the virus. So 70% more of infection. So I think this is a serious concern, and that is why you must be looking at how United Kingdom has taken very serious measures. And I think in India, we should take it very seriously and take measures, the standard measure that they've been taking so far, and don't go into complacency, and also reduce the indiscriminate, rampant use of these two medicine that are happening in the country because of pressure of pharmaceutical industry, whatever the reason that I'm really worried about that. Yeah.
Right. Many thanks, Dr. Ram Vishwakarma. Very important point you have raised here that, of course, there is, we should not be getting alarmed unnecessarily, but we need to study the situation as it can become a little worrisome. The authorities in UK have taken quick measures to deal with the issue. Now, Dr. Balramji Omar, if we talk of the efficacy of the vaccine, now as the mutant is the variant of the virus, yet the genetic information it has is little different than the parent virus. So will it affect, theoretically I'm asking this question, theoretically, does it affect the efficacy of any vaccine when it comes to application on the virus?
If any vaccine is developed towards the spike proteins, specifically for the spike protein, that epitope is focused to the spike protein, it will definitely be inactive for these viruses. But most of the vaccine, I jotted it down, I studied, they are the DNA vaccine or mRNA vaccine or the whole genome vaccines. They won't have any effect regarding this change. So theoretically, it won't affect much. It will have some or maybe minor effect might be there, but it won't affect much theoretically.
Right. So as your opinion is that there won't be any impact on the virus. Dr. Anil Gurtu, the change of genetic sequence of the virus, does it have any efficacy on the vaccine? If we talk of coronavirus now, we have 3 vaccine candidates in India, and we have several other candidates around the world. And of course, the trials has already begun. So, what is your opinion? Will it have any impact on the efficacy of vaccine?
See, right now, we do not have the hard data. All this that has come about, this detection of this latest variant is based on mathematical modeling. These are computer mathematically modeled things which say it's 70% more or less transmissible and all. These are not data that has come from the clinical field or even the lab studies. All mathematical modeled, which creates a sense of concern, not amounting to alarm, as Dr. Ram very rightly
Yes,
said.
absolutely. Yeah, yeah. Dr. Ram
Now,
Vishwakarma was, as he also
he
mentioned that.
clarified, and the second point I wish to highlight, which I completely agree with him as a clinician, that overuse or rather misuse of these two drugs, favipiravir, which has no evidence as of now, or even remdesivir, which has no proven survivability or survival advantage over anything. So not doing anything and managing with oxygen and dexamethasone, the moderately severe cases, is the best way to prevent resistance from emerging. Now, as Dr. Ram very right, I fully support him. In fact, I followed that strategy myself.
Okay.
This vaccine and its concern. Now, let us be very clear that this spike protein is not one monolith spike. It is not that one homogeneous spike of one protein of antibody resistance forms to that, and that's it. No, it has components and subcomponents. To begin with, it has three huge pillars which conjoin together to form what's the so-called spike protein. Within that, there are domains, and there's S1 above and S2 below, and within them, there's several other molecular rearrangements. So, of them, so many of them, only 2 or 3 against them, there is some possibility of this mutation developing resistance to, which still does not render the whole vaccine against the whole of the structure, genomic structure, which is far more complex and heterogeneous to develop resistance against. So my understanding, I may be wrong, but my understanding is, from whatever little I know about molecular and genetic biology, is that just development of 2 or 3 domains will not confer resistance to the whole vaccine.
Okay.
RBD is still safe, broad and large, and RBD is just one small component of the entire spike protein. So as of today, I would say the vaccine is still safe, theoretically speaking. Concerns can arise if we misuse these drugs and vaccines. One person takes one dose, doesn't take the other, that way. Then what will happen is that this virus will develop intelligence. Virus is more intelligent than humans. It has intelligence. It does not have consciousness. So that intelligence is far more superior to us, and it can do it fast. And then that will create resistance. But if we stick to, as Dr. Ram said, our 3 broad social strategies of masking and distancing and avoiding closed space crowdings, and take the vaccination properly in both the sequences and not take one and then vanish away, and not misuse, or rather not use these antivirals at all, given what we know of the antivirals' efficacy, then we are in for a safe and, I suppose, an okayish period with the vaccines. There's nothing to get alarmed.
Right. So there is nothing to get alarmed, as Dr. Anil Gurtu puts it, but we must follow the COVID-appropriate behavior and also certain medical advice that Dr. Anil Gurtu was giving to us. Dr. Balram G. Omar, I know it's fairly early days, and that's what even experts. I'll just come back to you, Dr. Balram Omar. We have got back with Dr. Ram Vishwakarma here, sir. Sir, I know it's early days still in terms of analyzing the virus here, the mutant, I mean. How long will it take for us to actually understand whether this virus is going to have any impact on the vaccines? How much more data would be required over a period of time? Dr. Ram Vishwakarma?
Yeah, so I think that effort has already started. If you are following what is happening in UK and Denmark and other countries, people have already started looking at the segregating patients. In fact, there is one hypothesis going around, which is, according to me, scientifically not correct, that when you start vaccinating people, in fact, vaccines also create selective pressure for virus to mutate, because vaccine provide threat to virus and generate antibodies, and then virus will try to get out of it. So effort has already started in UK to segregate patients which are on vaccine, which are on placebo, or not receiving vaccines, and they are doing sequencing of those people to look at whether any of these mutations, out of these 9 mutations, any of them existing there. And I think I see forward that with this episode and some other episode which are appearing from South Africa in the same week, actually, there will be effort by pharmaceutical company to initiate parallel trials to actually, which are anyway planned for these vaccines for larger scale trials. But in trials, we take this factor in the mind because, as you know, so far we have been solely focused on spike protein from therapeutic point of view, for vaccine point of view. And I think now we have to sit back and look at, perhaps we should look at other targets. And one more thing I will add you, that the diagnostic regime also will have to be relooked. If we are using RT-PCR and we are using a primer where these mutations are happening, then RT-PCR will not work. So far, anybody who's using RT-PCR, so UK has issued advisory that any RT-PCR using primer for S1 protein should select some other primer. So there will be realignment in diagnostic strategy also, and then the trials of the vaccine should take in this. And also, more importantly, some of these monoclonal antibodies which have been recently approved, you must be hearing about it on antibodies or Eli Lilly antibody, and these antibodies are also targeting the same spike protein, neutralizing spike protein. So I think they also will have to be relooked at in terms of the dosing, how much you give. So dosing schedule of those will have to be sort of looked at. So I think this is a important message that we are getting, which I think all viruses give in their evolution. That there is no space for any complacency, no space of lowering our guard in terms of what measures we are taking, and being very, very careful in terms of indiscriminate use of these chemical drugs. That particularly country like India, I'm really concerned because outside India, much more restriction on favipiravir, mind you, is not approved anywhere other than India for. So I think we have to be a little bit more careful about it. Yeah.
Right. Absolutely. Dr. Balram Omar, as we are about to end the show, sir, your closing remarks. I mean, as Dr. Ram Vishwakarma also mentioned about the approach of the testing. Now, I mean, I know it's very crucial to pursue smart testing and tracing the track and spread and contain the variant. Do we require some changes in that approach as well, so as to prevent this particular mutant to impact or affect the vulnerable communities? I'm talking of India-centric issue here, sir.
First thing is that ICMR has already started a consortium for sequencing these variant strains in India also. They have started this project. Second thing is that, as Dr. Vishwakarma has said, that the type of genes which are used to diagnose this virus, they have to be looked into, or those kits should be avoided which are showing those variation. Right. If those will be used, it will show the RT-PCR test negative. So genes to be chosen, the 3 genes of different regions other than the spike proteins may be chosen. So it won't be affecting the real-time test, right? And third thing is that we have to also focus upon the antigen test also. We should choose those antigen tests which are recently been updated by ICMR also and by FDA also, which are more sensitive and specific. They may be an alternative, but may not replace the RT-PCR, right? This is the testing strategy. Third thing, mutation will go on, and we have to keep those golden approach of precautions.
Right.
If anybody is following the golden parameters of precautions, he will succumb. And in my view, there is no what do we say, leher. It's just a matter of time. It is always there. It will go on till the virus is stabilized, or such mutation there which makes the virus stabilized.
Right. So, well, many thanks, Dr. Balram G. Omar, Dr. Ram Vishwakarma, and Dr. Anil Gurtu for your time and extremely valuable opinions and inputs that you've given to us, and of course to our viewers, in having a better understanding of this mutant and how we should go ahead in such a scenario. Well, many thanks for our viewers as well. But before we leave, a small appeal by RSTV to all of you. Please follow COVID-appropriate behavior. Keep wearing face mask. Keep washing your hands and maintain the physical distance, or to say social distancing. These are the only things that help us in defeating the pandemic. Many thanks for watching this edition of show. Thank you.
Watch the